Thanks a lot. Hello, everyone. My name is Neira Dedić, and I'm working PhD at the Department of Botany and Zoology under the supervision of Professor Shinkova. And today I will briefly present the first preliminary results under the title of Impact of Viral Infection of the Immune Defense in Hybridizing Fishes. The objective of my study is to compare transcriptome profile of head kidneys between parental lines and hybrid lines to reveal immune genes acting in the response against pathogen infection. And we hypothesize in F1 generation hybrid vigor and the hybrid breakdown in post-F1 generation. Interspecific hybridization have a great potential for application in the aquaculture industry. It's mainly applied with the goal to obtain advantageous traits in hybrids that will improve breeding and increase production, which has been widely documented in fish. However, mechanisms which are underlying hybrid resistance or successibility are weakly known. Hybrids of F1 generation are frequently characterized by the hybrid vigor and genetically based mechanisms responsible for the growth, superior or better survival, higher resistance to parasites of F1 hybrids in the fish, and it have been recently investigated. Despite of the advantages arising in F1 generation, however, post-F1 generation in hybrid show reduced fitness due to the genetic incompatibilities, and this is known as a hybrid breakdown. On the left photo, you can see the ancestral population in red and the nuclear genotype AABB. When the population is split into different mitochondrial mutation in blue and red are fixed in each population. If the new mitochondrial DNA carries deleterious mutation that compromise host fitness, and the nuclear genome may evolve complementary mutation to restore the host fitness. When two populations contain co-adapted mitochondrial and nuclear mutation, encounter each other and generate hybrids, unmatched mitochondrial and nuclei cause the hybrid breakdown in F2 homozygote diploids. Also, in the middle photo, you can see that co-adapted genes complex should be disrupted when an organoline from one lineage is transplanted into the nuclear background of different lineage, and the degree of disruption should scale with the time of divergence. At part of B photo, you can see at the molecular level that co-adaptation predicts that mutation in one interacting gene will be compensated by mutation in its interacting partner. Also, on the C part, you can see the structural properties of the interacting nuclear and mitochondrial and cold subunits of cytochrome oxidase protein show different amino acid substitution patterns of contact, which is present in red, and versus non-contact amino acid residues, black, blue, and green surface. And the last part, it's considered as a mitochondrial interaction accounts for the low fitness of F2 generation and all hybrids later. Then back crosses of paternal and maternal parents line will yield different results. By reestablishing the few nuclear mitochondrial DNA genomic complement of parental line, only the maternal backcross is expected to result in wild type fitness. The high fitness of F1 hybrid indicate that co-adaptase acts as dominant alleles. Also, for this part, Ellison and Burton suggested and highlighted the role of maternal inherited mitochondrial genome in hybrid breakdown, and showed in the marine copepod that maternal backcrosses outperform paternal backcrosses concerning their fecundity, ATP production rate, or survival metamorphosis rate. When considering parasite load, it's an important measure of host vigor and may reflect either hybrid heterosis or hybrid breakdown. We use spring viremia of carp associated with common carp to examine heterosis of versus breakdown in hybrid of the fish specimens, common carp and gibel carp. Spring viremia of carp caused by virus carpio bullet-shaped RNA virus affect almost exclusively cyprinids. Primarily common carp, this environmental and economically important disease with significant outbreaks in culture and wild living fishes has been documented in Europe, America, and Asia. It's mostly widespread in aquaculture, causing high mortality and morbidity at the temperature between 10 till 13 degrees. And when other cyprinid species exhibit varying susceptibility, concerning Carassius species, they should exhibit some susceptibility to spring viremia of carp, as monoclonal antibodies against spring viremia virus were previously detected in Carassius auratus. It's really important to tell the DNA vaccine exists, but it's not implemented in aquaculture and wildness. When presenting my model of fish, there are two evolutionary closely related cyprinid fish. Common carp and gibel carp are widely distributed through the Central Europe. Common carp, it's a commercial important fish in the aquaculture of Czech Republic, while gibel carp is invasive species common in the hydrobiological system of many European countries, and it's often reported to enter breeding pouches of common carp. Specimens of common carp and gibel carp and their naturally occurring F1 hybrids were collected from the Hlabovetsky fish pond here in Czech Republic and transported to the facility in breeding. The hybrids were identified using metric traits and molecular markers, and all fish were separated according to the sex in two different tanks and simulated for ovulation or for the spermiation by carp purity and the susceptibility increasing the water temperature to 23 degrees. As a result of this step, as a result of artificial breeding, we produce 9 lines of offsprings, pure line of Cyprinus carpio, pure line of Carassius gibelio, 2 lines of F1 generation, first F1 line with Carassius gibelio mitochondrial DNA, and F1 generation with Cyprinus carpio mitochondrial DNA. After it, we have 4 lines of back crosses, including 2 lines of maternal back crosses, as you can see on the right side of this schema, and also 2 lines of paternal back crosses, back cross Cyprinus carpio and hybrid, and back cross hybrid and carpio. And after it, we had the F2 generation, both parental being representing of the F2 generation, F1 generation with Cyprinus gibelio mitochondrial DNA. After it, we infected fishes with spring viremia of carp. We sampled the tissue. We extracted the RNA. We have library preparation, sequencing, transcriptome assembling, and then we performed functional and differential analysis. Here, I just would like to tell that we obtained the highest number of transcripts for the Cyprinus carpio, and also that we obtained the highest number of up or down regulated genes in pure Cyprinus carpio, then in F2 generation, and later we have also following all those fish generation. When going deeply and investigating immune-related pathways, I would like to tell that this graph is showing poor lines with F1 generation, and most of pathways are up or down regulated after infection. I would like to tell that, for example, toll and indie signaling pathway or node-like receptors are down regulated, while the others we found that it's up regulated. This is concerning same pathways, but different lines, also pure lines, comparing with back crosses and F2 generation, where you can see that toll-like receptors or node-like receptors are upregulated, which was differently expressed in F1 generation. After it, we selected few pathways for preliminary results. like receptor signaling pathway complement and hemokin signaling pathways. You can see that most of them are up or down regulated, and from them we selected few interested genes. The first gene is carp virus induced gene. It's biomarker. It's a gene which is applied in the which is activating when would like to stop the viral infection. And as you can see, the highest level of expression was in a poor Cyprinus carpio, the first row, and the last as in F2 generation. The next gene which we selected is interferon stimulated gene 15. And in these genes, you can see that second row red point, pure Gibelio, and after it, two following F1 generation show a bit lower level of upregulation, while at the left side, poor Cyprinus carpio, and after it back crosses and F2 generation show a bit upper level of regulation, these immune genes. After it, there is one hemokin which is downregulated, and this hemokin we also found that in poor Cyprinus Carasus Gibelio, second row, after infection, and the third and fourth row, also F1 generation, show similar pattern of reaction of viral infection, while the pure Cyprinus carpio and post F1 generation have the similar pattern to reaction to our hybrid breakdown. And the last gene which we selected for the preliminary results, it's the gene of plasminogen activator urokinase receptor. This gene is activating like almost double in all infected groups, but you can see that it's a bit lower expressed in the second, third, and fourth, which we expected also as a hybrid vigor. By all those, I can conclude that hybrid of the F1 generation were characterized by hybrid vigor. Also, F2 generation back process exhibit or may exhibit reduced fitness to viral load, which will be hybrid breakdown. It seems by these preliminary results that Cyprinus carpio exhibits similar paternal immune response as a backcross generation, and also it seems that Carasus Gibelio and F1 generation show the similarity in the immune response to the spring viremia virus. I would like to thank you for your attention. Also, I would like to thank to all my colleagues for support and help, and also my supervisor, Andrea Veteshnikova-Shinkova. Now I'm open for your questions.
Splendid. So thank you very much for, again, complex and entertaining talk. And we are waiting for questions in chat. Are there any questions on the